Brain-Derived Neurotrophic Factor, commonly known as BDNF, is often referred to as the fertilizer for the brain due to its vital role in supporting neural growth and function. This protein is essential for the development, maintenance, and adaptability of the nervous system. BDNF not only helps neurons survive and thrive, but it also plays a key part in strengthening connections within the brain. Research has shown that BDNF can enhance both vision and memory by promoting the plasticity of neural circuits, particularly in areas like the visual cortex. Its influence extends to improving visual acuity and supporting cognitive functions, making it a central focus in studies on brain health and recovery. As scientists continue to uncover the potential of BDNF, it stands out as a promising factor in therapies aimed at restoring and optimizing brain performance. Brain-Derived Neurotrophic Factor, or BDNF, is a protein that plays a vital role in the nervous system. It supports the growth and survival of neurons, ensuring that neural connections are properly established and maintained. Within the visual cortex, BDNF is essential for both the initial development of visual pathways and their ongoing maintenance throughout life. This protein also promotes plasticity, which is the brain's ability to adapt and reorganize in response to new experiences or injuries. By facilitating the maturation of neural circuits and enhancing synaptic strength, BDNF helps the visual cortex remain flexible and capable of recovering from disruptions. Its influence extends to regulating critical periods of development, making it a key factor in shaping how we process and interpret visual information. Recent studies have revealed that brain-derived neurotrophic factor, or BDNF, plays a pivotal role in supporting the recovery of visual function and enhancing visual acuity. In experiments with adult amblyopic rats, non-invasive intranasal administration of BDNF led to significant improvements in visual abilities, including sharper vision and better depth perception. These effects were long-lasting and depended on the activation of specific signaling pathways in the visual cortex. Additionally, research involving transgenic mice demonstrated that increased levels of BDNF can accelerate the maturation of visual cortical circuits, even compensating for delayed development caused by lack of visual experience. Collectively, these findings suggest that BDNF could offer promising therapeutic strategies for adults with impaired vision. However, further investigation is needed to determine its full potential in human visual rehabilitation. In 2018, researchers investigated the effects of intranasal administration of brain-derived neurotrophic factor, or BDNF, in adult rats with amblyopia, a condition that models human lazy eye. By delivering BDNF non-invasively through the nasal passages, they observed a marked increase in BDNF levels within the primary visual cortex. This elevation was associated with significant improvements in visual acuity, ocular dominance, and depth perception in the treated rats. Remarkably, the recovery of visual abilities was long-lasting, indicating a sustained benefit from the intervention. The study also found that blocking TrkB signaling, the main pathway through which BDNF acts, prevented these improvements, highlighting the critical role of BDNF in visual rehabilitation. These findings suggest that intranasal BDNF delivery could be a promising approach for restoring visual function in adults with impaired vision. Research has shown that BDNF treatment can lead to enduring improvements in visual function, particularly in models of amblyopia. The beneficial effects of BDNF are not fleeting. Instead, they persist well after the initial administration, indicating a lasting impact on the visual cortex. Central to these improvements is the TrkB signaling pathway, which acts as the primary receptor for BDNF in the brain. When TrkB signaling is pharmacologically blocked, the recovery of visual acuity and other visual functions is prevented, underscoring its critical role in mediating the effects of BDNF. This relationship highlights the importance of both BDNF and TrkB in promoting neural plasticity and functional recovery within the visual system. The findings suggest that targeting this pathway could be a promising strategy for treating visual impairments that were previously considered irreversible. In 1999, researchers conducted a study using transgenic mice to investigate the effects of brain-derived neurotrophic factor, or BDNF, on visual development. The findings revealed that elevated levels of BDNF led to an accelerated maturation of cortical inhibition within the visual cortex. This process is essential because it helps regulate the critical period during which the brain is most adaptable and responsive to visual experiences. By promoting earlier maturation, BDNF effectively shortened the window of heightened plasticity, suggesting that it plays a pivotal role in shaping how and when the visual cortex develops. These insights highlight the importance of BDNF not only in normal visual development, but also in potential therapeutic strategies for visual disorders. In 2003, researchers investigated the effects of brain-derived neurotrophic factor, or BDNF, on visual cortex development in mice that were raised in darkness and deprived of normal visual experience. Remarkably, they found that overexpression of BDNF could counteract the negative impact of dark rearing. Mice with elevated BDNF levels showed visual acuity, receptive field sizes, and intracortical inhibition comparable to those of mice raised in normal light conditions. This suggests that BDNF plays a crucial role in supporting the maturation of the visual system, even when typical sensory input is absent. The study highlights the powerful influence of BDNF on neuroplasticity and its potential to compensate for environmental deficits during critical periods of development. Brain-Derived Neurotrophic Factor, or BDNF, plays a vital role in supporting the development and resilience of the visual cortex. Research has shown that BDNF can compensate for delayed visual development, such as in cases where normal visual experiences are disrupted or absent. For example, studies involving dark-reared animals, those raised without exposure to light, demonstrate that elevated levels of BDNF help maintain visual acuity and proper neural function, even when typical sensory input is lacking. BDNF promotes the maturation of inhibitory circuits within the visual cortex, ensuring that critical periods of plasticity are preserved. This neurotrophic factor also aids in the recovery of visual functions following injury or impairment, highlighting its potential as a therapeutic target. Overall, BDNF acts as a safeguard for healthy visual processing, even under challenging or adverse conditions. The recent discoveries surrounding brain-derived neurotrophic factor, or BDNF, have significant implications for developing new therapies aimed at visual rehabilitation in adults. Research has shown that increasing BDNF levels in the visual cortex can lead to lasting improvements in visual acuity and depth perception, even in cases where visual development was previously impaired. Notably, non-invasive methods, such as intranasal administration of BDNF, have demonstrated promising results in animal models, suggesting a potential pathway for human treatments. These findings indicate that BDNF could help restore visual function by promoting neuroplasticity and recovery, offering hope for conditions like amblyopia that were once considered difficult to treat in adulthood. However, while these results are encouraging, further clinical studies are needed to confirm the safety and effectiveness of BDNF-based therapies in humans. If successful, such approaches could revolutionize the way we address adult visual impairments and enhance overall visual performance. While the potential of BDNF in promoting visual function recovery is compelling, it is important to approach these findings with caution. Most of the current evidence comes from animal studies, and the effects observed in rats and mice may not directly translate to humans. Before BDNF-based therapies can be widely adopted for visual rehabilitation, rigorous clinical trials are necessary to confirm both their safety and effectiveness in people. Researchers must also investigate optimal delivery methods and long-term outcomes to ensure lasting benefits. As the field advances, careful evaluation will be essential to fully realize BDNF's promise in human visual health. Continued exploration, will help clarify its therapeutic potential and guide future innovations in vision science. In summary, Brain-Derived Neurotrophic Factor, or BDNF, stands out as a powerful agent in promoting both vision and overall brain health. Research has shown its remarkable ability to restore visual function, enhance acuity, and support neuroplasticity, even in cases where development has been compromised. These findings suggest that BDNF could play a transformative role in future therapies for visual impairments and neurological conditions. As science continues to uncover new insights into the brain's capacity for regeneration and adaptation, staying informed about breakthroughs like these is more important than ever. The ongoing exploration of BDNF and related neurotrophic factors holds promise for advancing our understanding of the brain and improving quality of life.